the biomedical text mining part of this workshop. So, yesterday we did uh, several activities, and we basically followed this paper. So, we basically followed this paper here, wherein first we did the information retrieval from the NCBI, and doing some uh, text mining with the query builder available with the NCBI. And then we moved on to document classification and then building a word cloud infographics, uh, which through which we can make colorful representation of the corpus of a large number of papers in one figure. So this is a very different. This is really very different from the kind of text mining we are familiar to do. We are basically familiar to read and write. And in our mind, we do text mining without mentioning that terminology anywhere. But we actually do text mining. And from the libraries, we go through the papers, we go through the journals. Nowadays, we don't go to library, so we go through the online repositories and use our manual abilities to carry out text mining. And this is done more or less throughout our life. Throughout our life, we keep doing that. Now, through these R packages, we are beginning to see that how from a large numbers of papers at one go using relatively simple scripts, we can create figures representing the data present in these papers. Now, the example that I have shown you may not be the most appropriate example for each one of you, because each one of you think differently. Each individual thinks differently. And each person has his or her own ways and means of going through the data, and the same tools, which we saw yesterday, can be used in many different ways in order to produce the best of the figures and along with it a short write-up. It is really very amazing to see that. So, how this happens? This probably happens because of one main running truth in all of us. And that running truth is each one of us has made some observations all along our life, and we have also questioned based on those observations, and those questions and observations, and along with it, whatever thoughts are packaged together, they stay in our mind. And then one day, when a medium like this, a computer medium, comes in front of us, these thoughts then start flowing through this medium, and the result is what is there to see. Because each person thinks differently, there is no one standard way of doing these tasks. The tools are the same, but the ways of doing these tasks can be very different between person to person and vary from person to person. So, what we have described here in this paper is the most common principle of the data mining, which is certainly drawn on the experiences of many others who published before us, and then we proceed forward with these basic tasks. Now, yesterday we used the function search ABSL, meaning that whatever classification we wanted to do in the most general way by capturing all the different variants of word part, which is the root part of the word that gives branches to different words, actual words, like diagnose, diagnosis, diagnostic. So that example allows one to capture all the articles in which the variants of this basic word or the root word are present, used by the authors. So people write differently. You know, if I give the same question and I don't say how you should write, I simply say, you write whatever you think about the answer in your own words. You will be surprised to find there will be hardly any similarity, although the basic sense will be the same. But the way each person writes will be very different because the thought process is different. And that is the natural beauty. That is nature's beauty. So we have to appreciate that. Now, how we can use the function search ABST? So suppose we have a little more specific requirement. In opposite of what we had yesterday, today let us say we had a very specific requirement. And the specific requirement is that we wish to know what are the papers in diabetic retinopathy that are reporting research results on or research studies on microRNA. So microRNA is a regulatory RNA and it is a very important molecular species present in our cell that regulates the gene expression at the transcript level. That's microRNA. So we start with the command miRNA ABS and we use search ABST and our corpus is diab retino ABS and we have this include meaning that wherever those terms are coming, include those papers for me. You can also exclude, the arguments are available for that. So in the simplest form we have include and we simply write MIR. So the MIR part of all the miRNAs are common. So we write that and we press enter and we can see there are 360 abstracts for miRNA or MIR. As before, this is an S4 object. So we go to the abstract and let's see the first abstract. And here you can see that this paper is doing research, reports research study.
Excuse me, sir.
Ah,
So what do you mean by S4 objects?
yeah. S4 object is an object in R which has the property that whatever functions you write for the S4 object will be available for all the objects, R objects, data objects that you have written, that you have use with the same S4 class. So it's an advanced structure of R and it uses basically the C++ base and it has what are called slots and each slot can be either a vector or a matrix. Any sorts of combinations are possible. And then you can write methods or functions which will extract these data from those slots. So the slot operator is the at character. And when I typed the at character, you can see the three slots that were coming were journal, abstract, and PMID. So any object of this S4 class, I can use it for the functions that are available in this package. So I don't have to write again and again special functions for each of these data. I can directly use the structure of the S4 and I can proceed. So this is what is the S4 structure of the R. And you can see here that the MIR has been captured very specifically. So this miRNA has been used in this research, and in the conclusion we can see these findings indicate that MIR 1815P ameliorates Hg stimulated proliferation, migration, and angiogenesis of HRMEs, EACs through inhibition of VEGFA, which might prove a new target for the treatment of diabetic retinopathy. So, what are HRMEs? Those are human retinal microvascular endothelial cells, and Hg is high glucose. High glucose is the problem of diabetes. You can see one more. And here we can see that the miRNA is mentioned here. miRNA is also mentioned here, MIR75P. So that MIR is a common, uh, how should I say, common short form present in all the microRNA nomenclature. If such a thing is available, then for microRNA we can use like this. And for LNC RNA, there is no such thing as far as I know. So when the nomenclature will come for LNC RNA, then one can uh, use it to classify the abstracts. So now out of the 10,000 abstracts, you can you got it classified to the microRNA subcorpus of the whole diabetic retinopathy corpus. So this is how the search ABST works. Now, today I'll show you another type of data mining, which is called entity recognition, named entity recognition and normalization. So for this, the package, in the package, the function is available, which basically gets data from this place, which is called pubdater, and this is again created from the NCBI NLM. And here they have used machine learning, artificial intelligence based approaches for entity recognition. So this is not in R, but what I wanted to show you was that this is the site where they give data free. As you can see, the data is free and can be accessed interactively through a web browser and programmatically via RESTful API. So the function available in the package, it uses this RESTful API approach and extracts the data from many papers in batches. So this was developed by these authors here from NCBI, and this is the title of the paper, Automated Concept Annotation for Biomedical Full Text Articles. So let us see and have a feel for what this is about. So we need to enter some terms here. We can enter the disease terms that we have, or we can enter the PMIDs that we have. So we have miRNA ABS. So we can ask for PMIDs in this way. So there are two PMIDs. So I'm just copying this and pasting it here, and we can see that immediately these two papers have come. Now we can click here. You can see in the title itself the miRNA is mentioned. And we can see here that again here in this title itself the miRNA is mentioned. And it is now mentioning the disease diabetes mellitus along with these cartoons. And here the full paper is also appearing. And you can see that the different entities are marked with different colors. So here the bioconcepts are given. So the gene is somewhat violet. These are genes. And then the disease is orange. So these are disease terms. This is the chemical term, glucose, iron, reactive oxygen species. They are chemical terms. Then mutation, there is nothing in mutation study reported here. Then species is also there, and human is a species. So that is also mentioned here. And if cell lines are there, then it would come. And you can scroll down below and you can see the different genes which are marked for easy uh, identification and recognition, a colorful approach. A similar thing here, it is not appearing. Let's see some more PMIDs. So we can see here another paper where we can see again it mentions about Ptis mellitus and the genes, species. miRNA is also considered as a gene. It's a very specific product coming from the different regions of our genome. And we can see here inflammation is again part of this disease. NF kappa B, MIR, and so on. So through colorful view, we are able to get this information. Now, here is a paper on exosomes or microRNAs in the treatment of diabetic retinopathy. So, exosomes are little organelles present inside the cell which have the capacity to process RNAs, the pre-RNAs. So, how exosomes, which are small vesicles, they do that is a interest. Most cells have this, and here what are marked are different disease or pathologies that are classified in these papers. This is the chemical and other terms like hypoxia, meaning less oxygen conditions, are also marked. And this marking is done by artificial intelligence tool that is developed by these authors of PubData. Here little statistics is also given. Diabetic retinopathy has been found 3 times, hypoxia has been found 2 times, and so on and so forth. So this is for view and read. This is for view and read. A pleasure kind of approach, which we can use and sort of happily read. If we had all the time in the world and no pressure on us to give any DAC report or fellowship enhancement report or thesis report, then this is surely a way to go. Pleasure. But pleasure is not always our line of work. She somehow avoids us. Therefore, we have to sometimes resort to some stressful type of work. Now, this is where the computer packages help us in order to relieve us from this stress so that we do little bit work and then we also get pleasure. So let's go to this R console again, and we can extract the same data from pub data. So let's say miRNA 50, and so you remember we were giving PMIDs and it was pulling out the papers. So we are going to do that here, but we are going to give 50 PMIDs, 50, in one batch. Why 50? Because the NCBI has put a limit. We don't know when that limit works also. It's a little bit mysterious. Sometimes people say it works, sometimes that limit doesn't work. So it depends on how many users are using that NCBI computers at a given time. So now we are in the evening. So in the Western countries, it is probably beginning daytime. So the students there will start hitting NCBI and then it will get overloaded. So if we are in the daytime, then we have some advantage. Then not so much request will come and maybe 50 can be changed to 100 sometimes. But I won't recommend it though. It's okay to do it 50 every time. And what we are doing here is we are using a L-apply approach, function of R. This is not function of PubMed miner package. This is function of R. And this is function of PubMed miner package. So you can combine the two functions for your work. In the case of search ABS and all, we were using only the PubMed miner function. But here we are combining the base R function with the PubMed miner function. So this is the beauty of R. That is why it's called a functional programming language. Functions and functions and functions. And you can keep going like this. So what does lapply do? It's like a loop only. It's a looping sort of a function, but it runs in a vectorized mode. So it's very fast. That's the only difference between lapply, and then it outputs the data in the form of a list. You might have heard in the Python class, the faculty who was giving that class mentioned about list. So R also has this list, which can take different types of data types. And you might have been introduced in the R class also. So we press enter and it's taking some time because it is bringing this data from NCBI. So a few seconds, I think, will go in this process. And we are going to get data for all these items that we saw: disease, genes, chemical, species. All this will come here in this list object. And the code in the pupdater function, it uses the RESTful API code and extracts that. So how this happens is there is a program from our end, and then this connects through that API code to the pupdater site thousands of miles away. And there another program runs and gives the output, which this fellow receives and packs it into the list. It's beautiful actually, how such interconnection is happening through the computer. Many things are happening. There is a pipe and there is a, host connection. All these things are happening in the computer technology terminologies. For us, it is a simple one line script that is doing this job.
Excuse me, sir.
Yeah,
Sir, function of PubMed minor over here is what? Actually, function of R is to lapply and function of PubMed minor is?
pupdater function.
Okay. And sir, actually, I was downloading the PubMed minor over here in our package. Actually, it was not able to download.
You don't have to download. You
So
go to packages.
it was not given over either.
Yeah, you go to packages like this, click on install, and here you type pubmed.miner.
Sir, I did this thing. Actually, I was getting PubMed mining. I was getting not PubMed minor in my R studio.
I didn't even type the full name and see pubmed.miner is coming already.
Sir, yours is coming, but in my system, it was
Not
not
coming?
coming.
It's not possible.
Actually, my version may be old one. I think so. R Studio.
Old one?
Maybe some kind of issue. That's why it was not able to install.
Sir, maybe it needs an update. Maybe that's why she's unable to.
Maybe
Okay.
that's okay.
So this is the version I am having, 2022.07.1. I have used older versions of our studio also. I've been using this for some years now. Because I have taught many students,
So maybe it need
batches.
some installation over there. That's why it is not able to download. Maybe some kind of problem is there. That's why I will try it again. Okay, sir.
Yeah, sure. Let's get one more, I think. One or two more, maybe. Now, the next one has to be 100. And 100 means actually 51 to 100. Rest everything stays the same. So, How many abstracts are there in this? There are probably 360. 360 is not a big number. Let's see how much we can do. Let's do 150 at least. Done. $150. It is a wonder that in such a short time, within couple of minutes, 150 papers data we have got. Okay, done. Now, these are different list objects, but we want one table. So there is another function in the PubMed miner called pubtator underscore result list to table. And we'll use the function C. We have used this function C in the search ABS also, if you remember. This is a R function, not PubMed miner function. It's a R function to concatenate. So let's concatenate this miRNA 50, then miRNA 100, and miRNA 150. These three data objects we are concatenating into this, and after concatenating it will run this function and give an output. So the output will be a file called result.txt. So that output has come. Now we want to open that output. We can open it using Excel. So I have Excel here. Click on Browse and go to Nextgenhelp. Click Open. It says there are no Excel files. So I use this option all files. Now all files have come into my view, and I go to this result and click open, and it says it is delimited. Yes, it is delimited. Next. And it is tab delimited. Yes, it is tab delimited. Next. And then click on finish. So this is a table of 150 papers. The data has come from PubData. PubData, after identifying the entities meaning genes, diseases, chemicals, and so on, they have all come in one table with these headers. The genes are in this column, the diseases are in this column, mutations, chemicals, species, and PMID also. So that if one wants to go back and look into a particular paper, then using the PMID, you can go back to that data. Now you can see that in the first paper, several genes are mentioned. This is the gene here, ZFAS1. Then this is the miRNA gene. This is ACSL4 gene, and so on. Now, by this greater than sign separator, there is a number associated with each of these entities. This number is called Antares ID. So I can now go to NCBI and I can go to gene and then paste this ID, this Antares ID. And click on search here, and the ZFAS1 gene entry comes up. And it is an official symbol given by Human Gene Nomenclature Committee. And it is an antisense RNA1. It is a non-coding RNA. It is also known by these other symbols, and it is representing a small nucleolar RNA host gene that produces a non-coding RNA. Increased expression or amplification of this locus is associated with cancer progression and metastasis. It may act as a molecular sponge for microRNAs. So, microRNA is a regulator, and then this gene produces an RNA which, as a sponge, it takes the microRNA cells. So that kind of complex transcriptional regulation is present in our body systems. Quite a wonder. Now, this Antares ID is really very useful, because if this gene is known by other forms and they are present in other papers, same gene, present in other forms in other papers, then we have to search and search and search and see what is this, what is this, what is this. So instead of that, this pupdater will map it to this single Anthres ID. And the moment it maps it to the single Anthres ID, then it is normalized. Meaning that through this ID, I can move forward and we can get the genes that have been reported in the paper, even though authors have used different forms. The practice of using this official nomenclature has increased now, but in previous years it was less, and people have a tendency to use the same symbol again and again that has been used before. They get sort of carried over, that carrying over sentiment is there. We are all susceptible to that. Therefore, in some of the conferences, those students who presented posters with this official symbol ID, they were given prizes. And some of the prizes, part of that prize covered some part of their travel, supper, and all that. Interesting. Truly interesting. So now more and more people are trying to use this, but what to do about the old papers? That will always be there as such in the NCBI. The NCBI is not going to edit those and bring it back to official ID. They don't have so much people and they don't have so much resources, and they're not going to make that effort, as far as I know. But they have provided a tool pupdater which can recognize them and map it to the same Anthres ID so that we can then use this data in a normalized way. So that's what is called data normalization in entity recognition in biomedical text mining. Let's see a little more of this. So we go back to our Excel file and now we go to the disease column. So in the disease column, you can see that the disease names are mentioned. And now after this greater than sign separator, we can see a short form like mesh, and then there is a alphanumeric ID. Alphanumeric means alphabet and number put together, like your PAN number. PAN number is alphanumeric, but not Aadhaar number. Aadhaar is only numeric, but PAN is alphanumeric. So in the same way here we have mesh ID, which is an alphanumeric ID. So what do we do with this ID? So we can use this ID to which it is mapped, and we can go to again the same NCBI, but click on mesh and click here so that it clears, and then press search. So it's a group of disorders under which this has come. This is the mesh. Mesh means medical subject headings. So this is again a standardized nomenclature. Standardized, I won't say much of standardized, but I will say organized into with proper IDs, the related categories. So you can see that the disease falls under nutritional and metabolic diseases, and this is called the tree also. Sometimes it's also called the trees. And the mesh ID that it has mapped to is within this diabetes diabetes mellitus. And here we can see that even other terms of the disease like diabetic complications, all of these come together. So the disease term which is mentioned by authors, diabetic retinopathy, is of course a very specific disease concerning the eyes of a person in a diabetic individual. And that is part of glucose metabolism disorders, comes under metabolic diseases, the entire classification taxonomy of the diseases. We can see from here. So those are the diseases. Then another one is the endothelial dysfunction. So let's look at that. Mentioned over there. But let's look at in the mesh here. So we click on this clear, the search bar, and then click on search again. So it is a corneal endothelial dystrophy type 2. It is the disease, or it is called altered morphology of the endothelium present in the eye. So this way, for each and every term present in the abstract, so the abstracts written by authors are very clear and very specific. They have used the terms which are scientific terms and medical terms in their text. So this also brings us to what is called proper scientific writing. So this is something we must learn, and we must include in our writing skills, that whenever we write about a disease or anything, the proper spelling, the proper way that it is mentioned, that has to be, and the meaning that is being conveyed is the same, that is being mentioned in the medical dictionary, or in the medical terminology, or in this medical subject heading, and not any other meaning that the author can themselves think or fuzzily write or anything. No fuzziness with respect to these terminologies. Go back to the same table, and in that same first paper we can see that glucose is a chemical that is mapped to it. Then there is iron, then reactive oxygen species, they are all mapped to this. Here again mesh ID is appearing. So let's have a look at it. And This is a beautiful example of normalization. You can see reactive oxygen species, somebody has written, that is mapped to D017382. And ROS is also mapped to this because often reactive oxygen species is mentioned by authors as ROS in capital, uppercase, not ROS in small case. That is the difference we have to note, and a lot of time is spent in editing papers because of these little things. Okay, and it is mapped to the same ID. Meaning is the same, so the ID is the same, but the actual words written look different. So this is called normalization. A beautiful example is here. So let's copy this ID and go to NCBI. Again, click on this clear search bar and click on search. And yes, we see reactive oxygen species, and it is also known by many other terms like oxygen radical or active oxygen and so on and so forth. Now in the mesh tree, reactive oxygen species is somewhere in the middle. Above that are free radicals, inorganic chemicals, and so on. And below that are very specific, more specialized hydroxyl radical, peroxides, then different types of peroxides, more and more specific they come down. Those are not mentioned in the paper. They could be, but they are not mentioned in the abstract text. So that is why they have not come. ROS authors have mentioned, so like that it has come. But in the full paper, they would have mentioned what type of ROS it is. Now, species is human, and you can see here only a number is coming. It's not an alphanumeric, it's a numeric. So you copy this, and for species, we can't go to mesh because it's not a mesh term. Wherever mesh was there, mesh word was also there in the pubdata data. Did you notice that? Mesh word was also there. So here mesh is not there. So we have to go to taxonomy, clear the search bar. Oh. And so those words are also mentioned here. And there are, under this, we can see, we can scroll down. So human is coming, human with H capital or H in small case. Then people are also mapped to the same taxonomic ID. Patients mapped to the same taxonomic ID. Mice is mapped to a different taxonomic ID, obviously. Mice is a different species. Human, again, there's nothing here in this paper. Then human patients, they are mapped to the same ID. Nothing here. Mice, mouse, they are both mapped to the same ID. You see, mouse is singular and mice is plural, and PubTater has been able to identify that. So that's the power of the artificial intelligence recognition tool. And when PubTater is giving this information in one go, it is running different tools. For gene identification, there is a separate program. For diseases, there is a separate program. Like this, different programs are there for each of these entity identification from the papers, and they are all run together, and then finally the results are collected together and output, and that output is being captured by the pubdata function in the PubMed minor R package, and then this table we can see using Excel. It's a tab delimited file, and we can see it using Excel.
Hello,
Yeah.
sir. I have one query that
Mm.
just the way you showed genes, diseases, mutation, and species, everything. In the same way, if we want to prepare this Excel sheet, you for any drug, say for example aspirin, and all the genetic related studies done on that in human or any other species. So can we get that table using these codes?
Yeah. So aspirin will be recognized by pubdata, I think. So some of these common common chemicals it will be able to identify. If it is something very unique or novel in which the updater chemistry engine has not been trained, then it will not be able to identify. Like phytochemicals. There are many phytochemicals which the puppeteer cannot identify.
Okay, sir.
So that we have to do it through a different approach.
Okay.
So yeah, that we have to do it through a different approach. But for the drugs and chemicals which are commonly known, a puppeteer will be able to identify because that would have been there in their training set. You can also read that puppeteer paper. They have given all the details, how they have done the work, and so on. And you can, it's a nice paper. You can go through it and have a feel for how they have done the work.
Okay.
So,
Thank you.
yeah, okay. So, what I will do is that we'll keep this file today as such, and tomorrow I will show you how in a batch-wise mode we can get more additional information from NCBI using the NCBI to ARC package. Okay, so we can do that tomorrow. And for today, I think this much if you do in your own work, it will be quite good actually. Because you do it and then try to have a feel for it and see what are coming and how they are coming. Okay. And then we can move to further data analysis using this data uh, after further processing this Excel file. If you do this work tomorrow's, at least some of you, and mail to me the text file or something like that, my email ID, uh, then I can also show it with your data, or I will show it with my data, whatever is convenient to you. And we can see what more we can get out of this data.
Sir, please share your email ID on the chat box.
Huh. So let me just okay. Yes. So my email ID is there, and you can note it down. So if some of you would finish it in a day, like how much time it took for 150 papers? Hardly uh, 1 minute or 2 minutes, right? So by the time you will have a cup of tea, or it can be easily done. So, and some of you may send it, then I can do it for you. Okay. And we can look at it and have a feel for it. And unless your data is very secret and you don't want to see others, then I will use what I have here. Shall we close for today with this? Any more questions? Acha,
Sir,
PPT.
can you please share your PPT?
Okay. In this chat, can it go?
Sir, it can be shared via email through which we are getting
Okay,
recordings every day.
you do one thing. You send me a mail from, I have given you my email ID. So you send me a mail, I will mail it to you. Uh, how else I can share? I don't know. Normally you can attach